X Variant 4 Good Manufacturing Practice Chapter 6 Quality Control Principle. This chapter should be read in conjunction with all relevant sections of the GMP guide. Quality control is not confined to laboratory operations but must be involved in all decisions which may concern the quality of the product. General, each holder of a manufacturing authorization should have a quality control department. This department should be independent from other departments. Adequate resources must be available to ensure that all the quality control arrangements are effectively and reliably carried out. The Quality Control Department as a whole will also have other duties such as to establish, validate, and implement all quality control procedures, oversee the control of the reference and/or retention samples of materials and products when applicable, ensure the correct labeling of containers of materials and products, ensure the monitoring of the stability of the products, participate in the investigation of complaints related to the quality of product, etc. All these operations should be carried out in accordance with written procedures and where necessary recorded. Finished product assessment should embrace all relevant factors including production conditions, result of in-process testing, a review of manufacturing documentation, compliance with finished product specification, and examination of the final finished pack. Quality control personnel should have access to production areas. Good quality control laboratory practice. Control laboratory premises and equipment should meet the general and specific requirements for quality control areas given in Chapter 3. The personnel, premises, and equipment in the laboratories should be appropriate to the tasks imposed by the nature and the scale of the manufacturing operation. Documentation. Laboratory documentation should follow the principles given in Chapter 4. An important part of this documentation deals with quality control. A specification, procedure describing sampling, testing records, procedure for and records of the calibration and qualification, a procedure for the investigation of out-of-specification and out-of-trend results, testing reports and/or certificate of analysis, data from environmental monitoring, validation records of test method. Any quality control documentation relating to a batch record should be retained following the principle given in Chapter 4 on the retention of batch documentation. Some kinds of data should be recorded in a manner permitting trend evaluation. In addition to the information which is part of the batch documentation, other raw data such as laboratory notebook and record should be retained and readily available. Sampling. The sample taking should be done and recorded in accordance with approved written procedure that describe the method of sampling, the equipment to be used, the amount of the sample to be taken, instruction for any required subdivision of the sample, the type and condition of the sample container to be used, the identification of container sample, Any special precaution to be observed, especially with regard to the sampling of sterile or noxious materials, the storage condition, instruction for the cleaning and storage of sampling equipment. Samples should be representative of the batch of materials or products from which they are taken. The sampling plan use should be appropriately justified and based on a risk management approach. Sample containers should bear a label indicating the contents with the batch number, the date of sampling, and containers from which samples have been drawn. Further guidance on reference and retention samples is given in Annex 19. Testing. Testing methods should be validated. A laboratory that is using a testing method and which did not perform the original validation should verify the appropriateness of the testing method. The result obtained should be recorded. Result of parameters identified as quality attribute or as critical should be trended and checked to make sure that they are consistent with each other. Any calculation should be critically examined. The test performed should be recorded and the records should include at least the following data: name of the material or product and where applicable, dosage for, batch number and where appropriate, the manufacturer and or supplier, reference to the relevant specification and testing procedures, test results including observation and calculation and reference to any certificates of analysis, dates of testing, initials of the person who performed the testing, initials of the person who verified the testing and the calculation. Where appropriate, a clear statement of approval or rejection, reference to the equipment used. All the in-process controls, including those made in the production area by production personnel, should be performed according to methods approved by quality control. Special attention should be given to the quality of laboratory reagents, solution, glassware, reference standards, and culture media. Reference standards should be established as suitable for their intended use. Their qualification and certification as such should be clearly stated and documented. Whenever conventional reference standards from an officially recognized source exist, these should preferably be used as primary reference standards. Laboratory reagents, solution reference standard, and culture media should be marked with the preparation and opening date and the signature of the person who prepared them. Where necessary, the date of receipt of any substance used for testing operation should be indicated on the container. Culture media should be prepared in accordance with the media manufacturer's requirement unless scientifically justified. Use microbiological media and strain should be decontaminated according to a standard procedure and disposed of in a manner to prevent the cross-contamination and retention of residue. Animals used for testing components, materials, or products should, where appropriate, be quarantined before use. Ongoing stability program. After marketing, the stability of medicinal products should be monitored according to a continuous appropriate program. The purpose of the ongoing stability program is to monitor the product over its shelf life and to determine that the product remains. This mainly applies to the medicinal product in the package in which it is sold, but consideration should also be given to the inclusion in the program of bulk product. In addition, consideration should be given to intermediate that are stored and used over a prolonged period. Stability studies on reconstituted products are performed during product development and need not be monitored on an ongoing basis. However, when relevant, the stability of reconstituted product can also be monitored. The ongoing stability program should be described in a written protocol following the general rules of Chapter 4 and result formalized as a report. The equipment used for the ongoing stability program should be qualified and maintained following the general rules of Chapter 3 and Annex 15. The protocol for an ongoing stability program should extend to the end of the shelf life period and should include number of batch per syringe and different batch size, relevant physical, chemical, microbiological, and biological test method, acceptance criteria, reference to test method, description of the container closure system, testing intervals, description of the condition of storage, other applicable parameters specific to the medicinal product. Stability. The product for the ongoing stability program can be different from that of the initial long-term stability study. The number of batches and frequency of testing should provide a sufficient amount of data to allow further analysis, unless otherwise justified. At least one batch per year of products manufactured in every syringe and every primary packaging type. In certain situations, additional batches should be included in the ongoing stability program. For example, an ongoing stability study should be conducted after any significant change or significant deviation to the process or package. Results of ongoing stability studies should be made available to key personnel and in particular to the qualified person. Out of a specification or significant atypical trends should be investigated. A summary of all the data generated, including any interim conclusion on the program, should be written and maintained. This summary should be subjected to periodic review. Technical transfer of testing method. Prior to transferring a test method, the transferring site should verify that the test method comply with those as described in marketing authorization or the relevant technical dossier. The transfer of testing method from one laboratory to another laboratory should be described in a detailed protocol. The transfer protocol should include but not be limited to the following parameters: identification of the testing to be performed and the relevant test method undergoing transfer, identification of the additional training requirement, identification of standards and samples to be tested, identification of any special transport and storage condition of test items. The acceptance criteria, which should be based upon the current validation study of the methodology and with respect to ICH, VICH requirements. Technical transfer or testing method deviations from the protocol should be investigated prior to closure of the technical transfer process. The technical transfer report should document the comparative outcome of the process and should identify areas requiring further test method revalidation if applicable. Where appropriate, specific requirements described in other European guidelines should be addressed for the transfer of particular testing methods. Thanks for your attention.